Good afternoon, students. In a series of EduCet lectures on immunology, I, Dr. Hardeep Kaur from Department of Zoology, Ramjas College, University of Delhi, would be presenting a lecture on antigens, a very important aspect of immunology. Antigens, as the students you all know, is a substance which, when introduced parentally into the body, parentally means it is not through the alimentary canal, that is not orally, into the body, stimulates the production of an antibody with which it reacts specifically and in an observable manner. Now, we can describe antigens as immunogens also, as tolerogens, as an allergen, and as in vaccine. All these are actually various different types of antigens we encounter. Of course, there is a marked difference between each type of an antigen. If we call it as an immunogen, there is a different property which it has, that is why we call it as an immunogen. So, if we call it as a tolerogen, it has a specific property. Now, if you again see towards the slide, this immunogen is an antigen that induces specific immune response. Now, when I say specific immune response, what I mean is that when this antigen is introduced into the body, it not only produces an immune response, that is, it leads to probably an activation of the lymphocytes, it can also react with the product of the immune response. For example, it can form a bond with the antibody, for example. Now, what would be the difference between an immunogen and an antigen would be? An antigen would be specifically that substance which can combine with an antibody, but in itself may not be able to lead to production of an antibody. But an immunogen in a strict sense is that antigen that not only leads to production of an immune response, but can also react with the product of the immune response. That is, for example, if it is an antibody, it can specifically react with antibody. So, technically we can say all immunogens are antigens, but all antigens are not immunogens, because all immunogens have to be antigenic. They will eventually react with the antibody and they will also produce immune response, but antigen need not produce an immune response. Now, in the same category, again we will see that tolerogen, as we can define tolerogen as the antigen that is capable of producing immunological tolerance, that is, immune system becomes unresponsive to this antigen due to prior exposure. And when does this exposure takes place? The exposure can take place during the development of the immune response. Certain antigens which are present in our body, the immune system, while during its development in an embryo or in the fetus, it is actually coming across those antigens, and now those antigens are no longer considered as foreign body. So, the immune system has actually become tolerant to this particular antigen. So, all those antigens that is capable of producing immunological tolerance, that means immune system has becoming totally unresponsive to this antigen, we call them as tolerogens. So, most of our body things which are already there or most of the body cells, molecules, cytokines which would already be there in our body, those all can act as tolerogen because our immune response is not going to react to this. So, all those molecules would be called tolerogens. Then comes allergens. Allergens are those antigens that causes a production of an allergic response. And finally, the vaccines could also be called a antigen because it is against, they are very attenuated antigens. We had already had a lecture on vaccines in which I had described various types of vaccines, in which vaccines could be an attenuated bacteria, attenuated microbe that actually leads to a protective immune response, a immune response which is going to help you eventually. So, vaccines could also be called a type of an antigen. So, these are the various types of antigens which we come across during the study of our immunology. Now, coming to the point where we come across immunogenicity versus antigenicity, as already described, I think the distinction between an immunogen and antigen should be very clear to you. An antigen is the one which is going to react to the immune response product, while immunogen is the molecule which actually would lead to production of an immune response and eventually reacting to the or combining with the product of the immune response. Now, what would be immunogenicity? Immunogenicity is the ability to induce an antibody or a cell mediated immune response, that is either the humoral response or a cell mediated response. While on the other hand, antigenicity is the ability to combine with the final product of the response. So, this ability we call it as immunogenicity or antigenicity. Along with if it is an immunogen, we call it as an immunogenicity, that is the ability to produce an immune response and to react to the product would call immunogenicity of that molecule, while antigenicity would be the ability to finally combine with the product of the immune response. That becomes, the ability becomes antigenicity. Now, as already said, most immunogenic molecules are also antigenic, but the reverse is not true. Now, coming to the classification based on antigenicity. Based on antigenicity, we can call it as an antigen as a complete antigen or an incomplete antigen. Now, a complete antigen is a substance which has both immunogenicity and immunoreactivity. By convention, we call complete antigen as an antigen. See, generally what happens in most of the books, the distinction between immunogen and antigen is not, and once they had described what is an immunogen and what is an antigen, they keep on calling even an immunogen as an antigen. So, distinction is missing, but a complete antigen is the one which is having immunogenicity as well as immunoreactivity, while an incomplete antigen is the one which has only immunoreactivity. Now, in this category, the haptens are very well known. Haptens are those molecules that can be made immunogenic. They itself are antigenic. They cannot lead to the production of immune response. However, they can combine with the product. That means haptens has the ability to combine with the antibody which is specific to it, but in itself it would not lead to the production of an antibody formation. But these haptens can also be made immunogenic by combining it to a carrier molecule. This carrier molecule could be a bigger protein or it could be any inert substance which itself does not lead to an immune response production. So, hapten along with the carrier, in certain cases this carrier is called a schlepper. Hapten along with the carrier can be made into a complete antigen or an immunogen. So, technically a complete antigen is an immunogen, and incomplete antigen is an antigen which can be called as an hapten also, or rather we can say hapten is a type of an incomplete antigen which can be made as a complete antigen by combining with a carrier protein or a schlepper protein. Now, here comes the real definition of hapten. Hapten has been obtained from a word called aptene, which means to grasp or to fasten. As it gets fastened or it grasps the antibody which is produced against it, it is not able to produce that antibody, but it is capable of combining to that antibody. So, it is a small molecule that is antigenic, but not by itself immunogenic. It can elicit an immune response only when attached to a large carrier such as a protein, which I called it as a schlepper also. The carrier may be one that also does not elicit an immune response by itself. See, we cannot have another molecule to which you attach this hapten and then that molecule itself become an antigenic molecule. We do not want that. This molecule's got to be an inert molecule. So, the carrier, generally proteins are those proteins which in itself are inert molecules. Now, antibodies can be made to haptens only if the hapten is covalently conjugated to a larger protein molecule called carrier. This concept was first of all introduced by Ulrich. Now, here is an example. If you see, the blue portion is the carrier molecule, the red triangular portion are small hapten molecules. Now, if you see the hapten carrier conjugate, if this is given to a rabbit, if a rabbit is immunized with a carrier hapten molecule, it will lead to production of 3 different types of antibodies. The antibodies to the hapten, antibodies to the carrier and antibodies to the conjugate of hapten and the carrier. Now, in this case only, if The typical example is dinitrophenol. This is 2,4-dinitrophenol. Phenol is a molecule which is a very good hapten. It can combine with the antibody which is produced against it, but on itself 2,4-dinitrophenol would not lead to production of the specific antibody. Now, if this 2,4-dinitrophenol is combined to BSA, that is bovine serum albumin, as a carrier, both of them can act as a hapten carrier conjugate and lead to specific antibodies which are produced against this dinitrophenol. So, hence the haptens can be made immunogenic in this way. Now, coming to the another way of classification depending on the source of the antigen, whether the antigen is your own antigen or it is coming from another species or coming from some other place. We call this we classify these antigens as Zeno antigens, allo antigens, auto antigens and heterophile antigens. Now, the first coming to the Zeno antigen, an antigen that is found in more than one species. The prefix Zeno means foreign. It comes from a Greek word xenos meaning stranger, guest or host. The pathogen could be a bacteria, virus, fungi, parasite, but is, for example, a antigen which is coming from altogether another species, we call it as a Zeno antigen. Now, for example, if bacteria invaded a human being, the bacteria becomes a Zeno antigen for us because it is coming from another species. Such antigens are called Zeno antigens. Now, there comes another category of antigen called allo antigen, an antigen which is present in allelic form encoded at the same gene loci or locus in different individuals of the same species. For example, when we talk about the ABO system of blood grouping, now for example, at the same loci, the antigen A could be there, it could be B, and in an individual, this individual could have only A antigen, the other individual can have only B allelic form, the other individual may have both of them, fourth type may not have any of them. So, at a particular locus in the genes of these different individuals belonging to the same species, we have different manifestation of this gene or different allelic forms of that gene at that particular locus in different individuals of the same species. Unlike the Zeno antigen, where it was coming from altogether different species, here the species happens to be the same. It is same Homo sapiens we are talking about, same human beings we are talking about, but different individuals have variety of allelic forms of a particular gene at a particular loci. That is called allo antigen. So, not only the ABO system of blood groups, but also the HLA system also comes under this category of allo antigens. Now, for example, if we have to do the blood transfusion, you have to ensure that the recipient and the donor have the same antigen at the same loci. For example, a person having a B blood group would preferably be given blood from a donor which is having a blood group B, because it would be a compatibility. You know, the loci is having the same gene. If it is different, for example, if you transfuse the blood in a recipient who has a B blood group from a donor who has a A blood group, the loci or the allelic form of the gene will be different, and that will act as an allo antigen in this recipient. So, there would be a problem, the blood transfusion would be a failure and would lead to problems later and would lead to hemolysis. So, such things have to be taken care of. So, in the same species, these two individuals, the A antigen is acting as an allo antigen for the person having B blood group. Similar goes for the HLA system, where tissue typing is done before a tissue transplant is accomplished. Now, coming to the next, that is, in case of autoantigen, in the next slide, you will see an antigen that despite being a normal tissue constituent is the target of a humoral or cell mediated immune response, as in case of autoimmune diseases. Now, in this case, the typical example we would like to give you is that most of the times, in fact, in almost all the times, the lens proteins or the eye component is never, the body is not actually gone through the tolerance for the lens protein. Now, if that same lens protein is used in as antigen in the same individual, that would lead to production of an immune response. So, release of sequestered antigen lens protein is released into blood to induce immune response, to induce inflammation of lens. The same protein, if given through some route intravenously or any as an antigen to the same individual, would lead to production of antibody against the lens protein, because these proteins had never, our immune system had never come across the lens protein during the tolerance. So, they had never developed a tolerance to these proteins. Similarly, the semen, the sperm of an individual can act as an antigen in the same individual. So, these are your own tissues, but because the immune tolerance has never been developed against that during the development, so these can act as a antigen to the same individual. Such antigens are called autoantigens. Of course, autoimmune disorders also come under this category when our body start producing antibody against our own tissue. For example, in hypothyroidism or hyperthyroidism, our body specifically, like for example in hypothyroidism like Hashimoto's disease, our body is producing an antibody against our thyroid tissue. So, that is a typical case of autoantigen where your own tissue is being acting as an antigen and our immune response is being produced against this antigen, which happens to be our normal body tissue. So, autoimmunities also comes under this. Another example is the denatured IgG. IgG is a normal antibody, but if it gets denatured, it becomes antigenic and it induces production of an antibody and it becomes a rheumatoid factor in patients suffering from rheumatoid arthritis. This denatured IgG is called a rheumatoid factor because it is acting as an antigen, an autoantigen, and the body is producing immune effect against this IgG, the denatured IgG. So, it is our own tissue, but body is producing an effect against it. Such antigens are called as autoantigens. Finally, we come to heterophile antigen. Similar antigens present on dissimilar organisms are heterophile antigens. Now, here we have closely related antigens which can occur on widely different organisms. For example, a blood group B antigen and the E. coli 086 protein, they both are very similar to each other. Although one is present in a prokaryote E. coli, the other is present on human RBC, totally different varied organisms, but they are so similar that you can have a cross reaction between them. Such antigens are called heterophile antigen. Another example being blood group A antigen and the pneumococcal type 14 capsular polysaccharide, they are also very similar to each other and hence called heterophile antigens. Now, coming to the factors affecting immunogenicity. Now, we have understood that what is immunogenicity, what is antigenicity. Now, if we understand what are the factors which affect immunogenicity, what are those things which should be present in an immunogen to make it a good immunogen. These are some of the parameters which our immunogen must have to become a good immunogen. If it is lacking in these parameters, then this immunogen may not be called a good immunogen and hence the body will not produce an immune response against this immunogen. So, for an immunogen to have a good immunogenicity or for an immunogen to produce a good immune response, such factors should be present in an immunogen. The very first factor, as you can see, is the chemical composition and heterogeneity. The second factor is the size, third factor is the foreignness, fourth is the susceptibility to antigen processing and presentation, and fifth is the degradability and solubility. Now, coming to the first factor, that is chemical composition and heterogeneity. Let us discuss each of these factors one by one. First factor is chemical composition. We must understand that an immunogen is a good immunogen if it is of a particular type. It has been found in nature that protein antigens or protein immunogens are better immunogens than the carbohydrate molecule as compared to lipids or nucleic acid. So, the best immunogens happens to be proteins or carbohydrates. So, we can say proteins are good immunogens because of their high internal molecular complexity and heterogeneity. We all know that proteins have a primary structure, they have a secondary structure, they have a tertiary structure, they have a quaternary structure. it's been found that if the molecule is big in size and it is heterogeneous in nature, it is having complexity, it becomes a better immunogen. Now again, it's already been found that if low molecular weight substances are there, which will come in the size, but low molecular weight substances will be more simplistic in nature, hence would be because of less of heterogeneity, they will become bad immunogens or not as good immunogens. Hence, a protein with the quaternary structure is much better an immunogen as compared to a protein with a tertiary structure or a protein with a secondary structure or primary structure. So, more the complexity in an immunogen, the better the immunogen is. Now again, if it's a homopolymer, let us say we have a synthetic peptides, we are using synthetic peptides to check the immunogenicity of these peptides. Now if we have a homopolymer, that means we are making shorter peptides of the same amino acids. We will find that the immune response would not budge, it would not act against these homopolymers. Bring up some copolymer, let us say 2 different amino acids, we had made a synthetic peptide, a longer peptide made up of 2 different amino acids, the immune response gets better. You introduce an aromatic amino acid, let us put up a tyrosine or a phenylalanine into this copolymer, the immunogenicity tremendously increases. Hence, the immunogenicity is not only dependent upon chemical nature, it is also dependent upon the heterogeneity. More the heterogeneity, better the immunogenicity. Next comes, the next parameter that comes is the size. The size of an antigen is equally very important. It greatly influences the immunogenicity. Immunogen with the molecular weight of 100,000 Dalton is supposed to be a very good immunogen as compared to a immunogen which has a molecular weight of 5,000 to 10,000 Dalton. So, bigger the size, more the complexity of immunogen, the better the immunogenicity of this immunogen. It will lead to a much better immune response. So, you increase the size. If a molecule is small, if an antigen is small, you can increase the size of an antigen by adding it to a carrier protein. Such carrier proteins or such molecules with which you can attach a small molecular weight size molecule could be a bentonite, could be choline. They are generally inert substances which are added to small molecules to increase their molecular weight. So, thing is that you have to have a bigger molecule, complex molecule to make it a better immunogen. The next factor which is important is foreignness. Antigens from related species are less antigenic or immunogenic than that of unrelated species. Phylogenetic distance between 2 species generate disparity between antigens. I will give you a typical example. If I take PSA, that is bovine serum albumin, it is coming from bovine, the cow family, and use it as an antigen in a cow, it would not lead to an immune response formation. But if I introduce the same BSA into a rabbit, it would lead to a tremendous amount of immune response. The reason being, the distance of the phylogenetic distance between a cow family and a rabbit family is quite a lot, and hence bovine serum albumin coming from the bovine family or the Bos family is less immunogenic in case of buffalo or a cow, but it is highly immunogenic in case of rabbit. So, the molecule got to be felt like a foreign, it is a new thing. The person, the immune response or the immune system should recognize it as a foreign material. It should not recognize as a related molecule, although it is not true for everything. Certain molecules can be used which have very similar to each other, can be used in unrelated species, but by and large, in most of the cases, the foreignness of a material becomes a very imperative factor for a good immune response. So, immunogen having a good phylogenetic distance, it is taken from a species which is phylogenetically much diverse from the recipient species, will have a much better immune response. It will elicit a much better immune response in the recipient. The next factor that comes is the susceptibility to antigen processing and presentation. Now, antigen presentation to T helper cells through antigen presenting cells takes place after processing, and this is essential for both humoral and cell mediated responses. Hence, molecules that cannot be processed and presented to T cells will will naturally become poor immunogens. The typical example I would like to give it here are the D polymers of the amino acids. We know that D amino acids or the dextrorotatory amino acids are nothing but stereoisomers of the levorotatory amino acid. The L amino acids are naturally occurring amino acids which we have in our body. Now, if we use a dextrorotating amino acid as an antigen or an immunogen, we will find that our body does not have enzymes to degrade these dextrorotatory amino acids. Naturally, if there is no degradation of these dextrorotatory amino acids, they cannot be processed. If an antigen is not being able to be processed in the antigen presenting cell, how will the peptides be formed? How will they go on to the surface of the T cell, APC or antigen presenting cell, and how would they be presented as an antigenic peptide to the T cell? Therefore, for an antigen to be a good immunogen, it should be susceptible to the antigen processing. If it is not susceptible to antigen processing and presented, it is like you process something and then present it to the T cells for the immune system to work upon it. Now, if it is not processed, not presented properly, the immune response is not going to be there. So, it will go either undetected or it will stay in the body, but it would not be, there would be no immune response to this particular antigen. So, this property is equally very important. Now, comes the degradability and solubility. This is the next property. The substance that is not soluble in body fluids fail to induce an immune response. If the substance is soluble in the body fluids, there is a possibility of exposure of hidden epitopes to induce immune response. For example, the pneumococcal polysaccharides are greater immunogens in mice, but not in rabbits, because the pneumococcal molecules are highly soluble in mice body fluids, but not in rabbits. Therefore, the antigen or the immunogen to be a good immunogen or to have a good immune response, it should be soluble in the body fluid. A immunogen which is not soluble would not be recognized by our immune system, hence there would be no immune response against that immunogen, as I gave you the example of pneumococcal polysaccharide. So, these 5 are the most important factors that affect the immunogenicity in an immunogen in an body system. The chemical composition and heterogeneity, the size of an immunogen, the foreignness, the susceptibility to antigen processing presentation, and degradability and solubility. These are the key factors. If an immunogen comes best, it fulfills all these parameters, it can be classified as a very good immunogen. Now, additional factors which are affecting immunogenicity are the genetics of the recipient, the genotype, that is the MHC, the dosage of the antigen. For example, if we are giving a very low dosage of an antigen, the body may not come across it, may not recognize the antigen. So, there has got to be an optimal dose of the antigen for the body or for the immune system of the body of the recipient to recognize it as an antigen. The dosage which can vary depending on what immunogen we are talking about, this dosage is very important. Again, the number of dosages of the antigen, because once the antigen has got into the body, after a while it will be degraded or metabolized. Now, after the first dose has been given, whether this antigen is going to stay in the memory in the form of the memory cells, or for example, the memory of this antigen will stay in the immune system, or it does not, or the antigen gets completely removed within few hours. It depends, it gives further rise to the number of doses, whether a booster doses of an antigen, for example, in vaccination, a primary vaccination is given, followed by booster 1, booster 2, etc. After number, let us say in the first month, 0, 1, 6 months, booster dosage is given of the antigen to again revive the immune response, again revitalize or again awaken the immune response against that particular antigen. So, the number of doses of an antigen is equally important. Again, the route of administration also plays a very important role in immunogenicity, whether it is an intravenous, it is subcutaneous, intraperitoneal, oral, or it is through inhalation. All these factors also play a very important role, whether there would be an immediate immune response, whether it will take much more time. For example, if it is intradermal, it might lead to slow release of an immunogen as compared to an intravenous. So, the route of administration also plays a very important role in defining the immunogenicity. Again, the use of adjuvant is another factor which is very, very important. What exactly is an adjuvant? Adjuvant, as you can see in this slide, adjuvare means, it's in Latin, it means to help. These are those helping compounds. In immunology, an adjuvant is a component that potentiates the immune response to an antigen or modulates it towards the desired immune responses. These immunological adjuvants are added to vaccines to stimulate the immune system's response to the target antigen, but do not in themselves confer immunity. For example, I will give you a tetanus, diphtheria and pertussis vaccine, for example, contains minute quantities of toxins produced by each of the target bacteria, but they also contain some aluminium hydroxide. There are many other examples of adjuvants like alum, incomplete Franz adjuvant, complete Franz adjuvant, bacterial lipopolysaccharide. But I guess you have understood what exactly an adjuvant is doing. Adjuvant is causing the antigen or an immunogen to stay for a longer time in a body. This alum, which is an aluminium potassium sulfate, it precipitates the antigen, resulting in its increased persistence in the body. It increases the size of antigen, hence increases the rate of phagocytosis of this antigen. Now, alum leads to a longer retention of an antigen in the body. Longer the antigen stays in the body, it does not get metabolized by the system, there are chances that it will keep on tinkering the immune response or the immune system, and a continuous immune response will be produced against this antigen. So, the purpose of adding an adjuvant as I gave you examples, there are incomplete Franz adjuvant, complete Franz adjuvant, complete Franz adjuvant has some mycobacteria. I can give you the composition. In incomplete Franz adjuvant, you have a mineral oil based increased persistence of antigen. In case of complete Franz adjuvant, along with the incomplete Franz adjuvant, you also add some dead mycobacterium. Now, the main purpose of adding adjuvant is to increase the retention of the immunogen or the antigen within the body. Similarly, bacterial lipopolysaccharides have also been used as good adjuvants for retention of an antigen inside the body. Now, what is an epitope or an antigenic determinant? After having understood an antigen, let us come to the point where we understand what is an epitope. The entire antigen could be a big molecule, or the entire, I would be calling antigen immunogen, constantly we keep on changing the nomenclature, we keep calling immunogen as an antigen, and as you can see in many books. But an antigen or an immunogen, if it is a big molecule, the entire antigen or an immunogen is not going to elicit an immunogen. It is the specific regions of an immunogen that leads to the response, immune response. So, it is that region of an antigen that binds to a T cell receptor, that is TCR, or a B cell receptor, which is generally an antibody. Since an epitope is the part of an antigen that binds to B cell or TCR, it is that part also that determines the antigenicity of the antigen. That means since it is that part where the B cell binds through an antibody or the T cell binds through a TCR, it defines what kind of an antibody will be produced against it, or what kind of a response would be. So, the response would be specifically to that region. That region which is actually binding to TCR or to the antibody in the whole molecule, that particular region we call it as an epitope or the antigenic determinant region. Naturally, we will find that it has been found in nature that T cells and B cells recognize different epitopes on the same antigen. Now, if for example, if we have a glucagon, which is a 29 amino acid long hormone, it has been found if it is given as an immunogen in a body in mice, the B cells generally react on the amino terminal, while the T cell generally reacts on the carboxy terminal, hence proving that B cells and T cells recognize different epitopes in the same antigen. So, what determines that this is a better B cell receptor or epitope or this is a better T cell epitope? That is influenced by various factors. So, various factors influence that these epitopes would be very good as a T cell or would be very good to tinker a T cell, or these epitopes would be much better to tinker a B cell or would be very good to produce an antibody response. Those factors are different. Now, here in this slide as you can see, in an antigen binding site of an antibody, here is an antigen and you can see the antigen is having 3 different types of epitope. It is a big molecule. Naturally, it will have various different proteins. So, different protein or peptides, if it is a peptide antigen, will have different configuration, chemical as well as spatial. So, these differences will lead to different types of epitopes. As you can see, the different epitopes on the same antigens are combined by different types of antibody or they are leading to production of different types of antibody response. For example, the A epitope leads to antibody A, the B leads to antibody B, the C leads to antibody C. Now, coming to the classification of antigenic determinants, the antigenic determinants or epitopes can be classified according to the structure of their epitope. For example, they are called conformational determinants or sequential determinants, or they can also be classified according to what they are leading to. So, they can be called, what response they are causing. They can also be called T cell epitopes or B cell epitopes. Now, coming first to the conformational determinants, antigenic determinants that are formed by amino acid residues that are not in a sequence but become specially juxtaposed in the folded protein. We all know that proteins have tertiary configuration, they have secondary configuration, secondary structures, tertiary structure, quaternary structure. So, in a tertiary structure, some amino acids will come closer to other amino acid because of the folding of the protein. Now, these amino acid coming closer to another amino acid which is in a linear molecule are not present one after another. So, that region can form a very good epitope or an antigenic determinant. Such epitope would be called a conformational determinant. Now, however, there are certain epitopes which are formed by several adjacent amino acid residues. That means, amino acid residues one after another which forms a linear or sequential determinant. That means, here the amino acids are arranged linearly 1, 2, 3, 4, 5. But in a molecule conformational determinant 1, 2 might be coming closer to 98, 97, 98 because of the conformation. The folding of the protein is happening in such a way that the 1 and 2 is coming closer to 97 and 98 and hence 1, 2, 97, 98 is actually forming an epitope. That is called a conformational determinant. This is because of the folding of the protein. While a sequential or a linear determinant is where the amino acids are coming one after another. Here as you can see in the diagram, the first diagram is of the naive antigen or the folded antigen, where there are, you can see, because of the folding of the protein, the conformational epitope which has come closer to each other, the amino acids which have come closer to each other because of the folding of the protein. While in the second one, the darker portion shows the linear configuration of the amino acids which leads to, this is obtained only after the native antigen has been denatured. If in a folded native state or the naive state, the antigen is in a folded state, the epitopes would be different, they would be called conformational epitopes. They have come together because of the folding of the protein. However, if we denature this antigen, the epitopes we get would be the linear or the sequential epitopes. Coming to the classification based on the immune response, we got antigenic determinants which are called B cell epitopes and the T cell epitopes. Now, what could be the antigenic determinant property that makes it a better B cell epitope? These are the properties which define whether this epitope will lead to B cell immune response. The first being the ability to function as a B cell epitope is determined by antigen binding site on the antibody molecule which is displayed by the B cell. That means, if we have a antibody binding site. We just described there is a, in our next lecture we will be dealing with our antibody structure. There is an antibody binding site where the antigen binds. Now as I just told you, the entire antigen is not going to bind. It is a small portion of an antigen which is called an epitope, which is going to bind in the antigen binding site. It is the antigen binding site of the antibody that determines which would be a better epitope, B cell epitope. For example, in this, the binding between the antigen binding site of an antibody and the antigen is a very non-covalent binding. It is a very weak binding. Now, using X-ray crystallography studies, it has been found that smaller ligands like carbohydrates, small oligonucleotides, peptides or haptens generally bind in the deep pocket of an antibody. For example, there is an angiotensin 2 which is an octapeptide, which if it acts as a antigen, it binds in the deep narrow groove which is formed in the antigen binding site of an antibody. Now in this case, since it is a small molecule, it just gets lodged in the antibody binding site, and it would be a very good binding between the two. However, if it is a globular protein, for example HEL, hen egg lysozyme. Now egg white lysozyme, HEL, which we call, it is a globular protein. Now if this protein acts as an antigen, in this case the antibody antigen binding site of an antibody does not bind in the form of a deep cleft. Like for example, this is an antibody, the antigen binds like this, but in this case what happens, they bind in a undulating manner on a surface level. The pockets of the antibody is just following the depression on the antigen. That means the depression of an antigen is going into the pocket of the, and the protrusion of an antigen is going into the pocket which is formed in the antibody binding site. So the globular bigger antigens would bind in a surface undulating membrane like a manner, while the smaller antigens will just got lodged in the, this all has been found using X-ray crystallography studies. So it is the antigen binding site of an antibody that defines how this immunogen is going to bind. The second property, as you can see, is that the most of the B cell epitopes are composed of hydrophilic amino acid. That means they are water loving ones. The next is that they can contain sequential, that is linear, or conformational epitopes. As already described, a good B cell epitope may be present as a, because of the secondary structure or the tertiary structure of a protein of the antigen. Hence it could be having a conformational, it could be a conformational epitope where the epitope is formed between such amino acids which in a linear molecule are far apart, but have come together because of the secondary or the tertiary structure of an amino acid. It could also be a sequential or linear epitope. So it can be both. It has also been found that most of the B cell epitopes are found on the flexible region of the immunogen. It's been found that if this flexible region ensures that immunogen just gets captured by the B cell. If it is not in the flexible region, or if it is in a rigid region, the combination between a B cell epitope and the B cell is not there. It is not generally been found to be there. So more the epitope in a flexible region, more the chances of it coming in contact with the antibody. It has also been found that most of the B cell epitopes may contain overlapping or non-overlapping determinants, and they are generally located on a topographically accessible region of an antigen. Now, if an antigen or an epitope is hidden inside the protein, it will not be recognized by an antibody. Most of the antibodies recognize those epitopes which are on an accessible region. If it is hidden inside, this epitope is hidden inside in the folded structure of the protein of an antigen, it will not be recognized, as most of the B cell recognize those antigens which are soluble antigens. So they are just moving in the bloodstream, and if this antibody will come across those epitopes which are just in an accessible region, they cannot explore inside, they cannot look at the epitopes which are hidden inside the tertiary structure of an antigen. So it has to be in an accessible region of an antigen. Now coming to the properties of a T cell epitope, they will be much different. Now in case of T cell, you must understand the antigen is taken up by an antigen presenting cell or a antigen is present in an altered self cell. Now this antigen needs to be processed, that is broken down into smaller peptides, and these smaller peptides are presented onto the surface of APC or the self altered cell, and it is presented along with the MHC molecule to the T cell, either the T helper cell or the T cytotoxic cell. So the first property is the antigenic peptides recognized by T cells form a trimolecular complex with T cell receptor and an MHC molecule. That means it has an epitope to react non-covalently with the T cell receptor and an agritope to react with an MHC molecule. Now if you see in this following slide, here is a protein antigen in the cell. This could be an APC. It has gone inside the cell. It has now been processed or broken down. Then it is being shown along with the MHC to the T helper or T cytotoxic cell. If it is a T helper cell, then we are talking about MHC2. If it is a T cytotoxic cell, we are talking about MHC1. Now, if it is a T helper cell, we are talking about an antigen presenting cell. If it's a T cytotoxic cell, we are talking about an altered self cell. Here what we are saying that the antigen is forming a complex wherein one surface of an antigen is the red dot shown here on the third slide. This antigen where it is combining with an MHC, that area is called an agritope. While an antigen combines to the T cell, that area is called an epitope or the area where it binds to the TCR. Now coming back to the other properties, the T cell epitopes are always presented with MHC. As I just told you, these epitopes will only be presented, it would be a trimolecular or a ternary complex wherein one part is the antigenic peptide, second is the MHC either 1 or 2, and the third is the TCR, the T cell receptor. Now, so this could be a CD8 receptor or CD4 receptor depending upon what we are talking about. But it could be, it's a T cell epitope is always going to be presented along with an MHC. It cannot go singly. It cannot be shown to a T cell singly. It has to form a complex first with an MHC and then this complex is shown to the T cell. So, antigen processing is required. It does not bind to soluble antigen. These are two properties basically. The antigen which has come inside an APC or a self-altered cell, this antigen if it is present, it has to be processed. The entire antigen cannot be shown to the T cell. It has to undergo processing. That means the breakdown of this antigen got to take place. Only then it will be presented to T cell. Now again, that means that the T cells do not recognize soluble antigens or all those antigens which are present in the bloodstream or moving in the bloodstream, which is very unlike the B cell epitopes. In case of B cell epitopes, the epitopes were in the accessible region and they were present in the soluble antigen which is moving in the bloodstream, coming across an antibody, and antibody by default coming in contact with the topographically accessible epitopes. In this case, it is programmed. Here the antigen has to be taken up by the APC, broken down, and only then these peptides would be shown to the T cell. Finally, the epitopes recognized by T cells are often internal peptides, which is very much unlike the B cell epitopes. In B cell epitopes, they were on the accessible region, just physically available region. Here or spatially available region. Here it was what is happening, even the internal peptides because of processing can become a T cell epitope. Here because processing is taking place, the breakdown of the antigen is taking place, the internal peptide can also act as a T cell epitope, unlike the B cell where no such processing takes place. So, the hidden epitopes remain hidden. They would never be recognized by the immune response or never be recognized by the B cell antibodies. But in this case, hidden epitopes can also act as good T cell epitopes and they are generally sequential in a native protein. Now, here is a comparison of antigen recognition by T cells and B cells. Interaction with antigen in case of B cells involves a binary complex of the membrane immunoglobulin and antigen, while in case of T cells it is a ternary complex. In case of B cells, the binding of soluble antigen takes place, in T cell no such binding takes place. MHC involvement is not essential in B cells, while it is very much essential in case of T cells. The chemical nature of antigens is generally protein, polysaccharides or lipids in case of B cells, while in case of T cells it is mostly proteins and sometimes lipids. The epitopes properties are, they have got to be on an accessible region, they got to be hydrophilic in nature, they should be mobile peptides, that means they should be on a region which has a high mobilization or flexible region. They can contain sequential or non-sequential amino acids. While in case of T cells, generally the internal linear peptides are produced by processing of the antigen and they are finally bound to MHC while presenting to the T cells. Now, there is another category of antigens called super antigen or SAGs. They are a class of antigens that cause non-specific activation of T cell, resulting in polyclonal T cell activation and massive cytokine release. SAGs are produced by some pathogenic viruses and bacteria, most likely as a defense mechanism against the immune system. Now, when we compare it to a normal antigen induced T cell response where only 0.001 or 0.01% of T cells get activated, the amount of T cells that get activated due to super antigens is almost 25% of the body T cells get activated. That means we have a hyperactivation of T cells. Most extensive studied SAGs is staphylococcal enterotoxin B. This is a new field which has come up in case of antigens, the super antigens. With this, I would like to end my lecture. Thank you very much. Hope it was of use to you. And in our next lecture, we will learn more about the antibodies and another important parameter of immune system. Thank you very much.